why are we talking about in a financial advisory conference the virus and vaccines? And the answer is, I hope that 2020 has made clear to everybody that these are all forms of capital. Our health capital in this country has a direct impact on our financial capital. The best performing stock market in the world this year, I think, is the Chinese stock market, up about 25% year to date. And that country has done a very, very strong job of containing the virus. And so we're seeing that there is a correlation between our health capital and our financial capital. And so that is why we are talking about these things. There is a direct link between them. So with that, let me go ahead and begin to introduce our first speakers. Dr. David Lee Thomas is going to speak first. He is professor of medicine at Johns Hopkins, director of infectious diseases, and the Stanhope Bain Jones Professor of Medicine at the Johns Hopkins School of Medicine. And he will be followed by Dr. William Bishai, who is the co-director of the Johns Hopkins Center for Tuberculosis Research. Dr. Bishai received his MD and PhD degrees from Harvard Medical School in 1989, did his internship and residency at the Brigham and Women's Hospital in Boston, and received fellowship training in the Division of Infectious Diseases, also at Johns Hopkins. And as we have seen in the last few days in financial markets, we have seen a wavering in the value of stocks. As we have come to understand that there is an uptick in virus cases, and also there have been, as far as I can see, some news articles recently and pronouncements from researchers that we may not be as close to a vaccine as maybe people were thinking. So we'd like to get some insight on that from our experts today. And then also the vaccine itself has become a focal point in the elections, in the presidential elections themselves, as there are pronouncements as to whether we're going to see a vaccine before the elections or immediately in the wake of the elections. So in this time when people are not listening to experts nearly as much as we should be, we are very, very grateful to have Drs. Thomas and Bishai with us today to give us as lay people a better understanding of what is a vaccine and how do we understand the push to get a vaccine in addressing the COVID-19 virus. Thank you very much for being with us. And I think, Dr. David Thomas, you're going to go first. So I will turn it over to you at this time.
Yeah, thank you very much, Leonard. We're delighted to talk with you a bit this morning, late morning, on COVID and COVID vaccine. Bill and I want to make sure we answer your questions, so we're going to talk in less than the total allotted time, and then make sure that you have an opportunity to ask us what you're especially interested in. So I'm going to begin with some general comments about vaccinations and the COVID vaccines in particular, and then Bill will give you some exciting innovative ways of looking into the COVID vaccine story. By way of disclosures, I have these activities, and I have no, and will be sharing no unique insights into this that aren't publicly, into this topic that aren't publicly available. Okay, let's start down. Sometimes, some of you probably know more about this than Bill and I, and for others, it's kind of a new concept. And so I'm going to begin broadly just with the idea of what is it that we are trying to accomplish with vaccines, because sometimes it helps to have a broader construct to build your knowledge base on. And with vaccines, of course, the idea is that we would introduce some element of an infectious microbe and train the body's immune system so that when the real microbe comes along, it's not for the first time that it's seen it, and it either completely prevents the body from being infected, or more often actually just prevents the disease complication of the infection. So the microbe might get into the body, but you don't get sick from it. And so that's the overall. And then when you see a broader perspective and consider vaccinations from a public health perspective, we're of course interested in what happens overall in the whole population. And that gets into issues of not only does vaccine get into someone's arm, and when it gets there, does it create the right immune response, but it gets into bigger questions of, do the right people who need the vaccine get them, and does the protection of one person then also prevent the spread to others and create things like what we call herd immunity that you hear about sometimes in the press. So that's sort of the general concept. And with vaccinations that you might be familiar with, this first business of completely protecting against infections, for example, with measles, that vaccine keeps the measles virus from coming into our bodies and helps us, for all intents and purposes, not be infected. And similarly with smallpox, it achieved that sort of sterilizing or completely preventing the infection. But oftentimes it's more the case that we're dealing with vaccines that attenuate the infection. The germ gets in, and yet the serious complication doesn't occur. And sometimes the vaccines, like for example, we give all the children in the United States this pneumococcal vaccine, and that vaccine given to children actually protects adults against getting pneumonia, because the kids don't carry the germ around, so the adults don't catch it from the kids, and at a population level, it can have that sort of success. And then others you might be familiar with, like the shingles vaccine. It's not to prevent you from getting the virus that causes shingles, because you got that when you were a kid, most likely. And instead, it prevents you from getting the shingles manifestation. So it's training the immunity to prevent the disease, not the infection. So with COVID, what are we doing? What's the goal here? So, and of course, we would love for the COVID vaccines to protect against infection, like with measles. But like with flu, we're not expecting that to occur. We're expecting that people may go ahead and get infected. We're really mostly, and the vaccine trials are designed to test this idea that the infected people won't get as sick. They won't get hospitalized, they won't get admitted to intensive care, and that it will have its impact on symptomatic COVID infection as a metric. And then, of course, we are hoping for this sort of population benefit, that we can get enough people protected around the world that it won't be a risk. Okay, let me see what I'm doing here. Okay, yeah. So now, one of the things that you read about in the papers is this, like, how do we know which part of the germ, in this case, COVID, to put into the body, and which one do we want an immune response to? Because some people thoughtfully ask, well, wait a minute, I thought that the whole problem with this thing was that you get a kind of exaggerated immune response, and that's actually what puts people in the hospital. So why would we want to prime the immune system and get it all ready to go? Wouldn't that maybe make things worse? And wouldn't it, couldn't that be a complication? And then how do you know which part of the virus to use and that sort of thing? And so we do have some sense of that from studies in animals, and those are important questions. We have a sense that the outer shell of this virus is the one that is going to be the one that we need to create immunity, antibodies against. And that's the one that at least the United States programs are betting on. And so we can measure those antibodies, and that gives us some proxy in the early phase of vaccine testing, whether or not it's working. And then, and we call those the anti-spike antibodies. And that's kind of you'll read about that, and I may even mention that later on. So in the United States, vaccine development goes through these, and as with drug development, goes through these pretty predictable stages that you're probably familiar with. When people talk about, oh, this vaccine is going into phase 3, with COVID, that means that in about 30,000 people, enough people are getting the vaccine or the placebo in order to evaluate not just whether it's safe, and not just whether it produces these antibodies, these anti-spike antibodies, but also to find out if it's working. And so why so many people? Why do you have to have 30,000 people? It's because even in the United States with the epidemic the way it is, not very many people at a population level will be infected. And so it's thought that, for example, you would need to have at an estimated incidence that is fortunately moderate and low, to have 15,000 people exposed in the vaccine arm and then 15,000 in the placebo. That will be necessary in order to get, let's say, 150 cases in the placebo arm. And that's the kind of robust study we would need to see if, for example, we could reduce by 50% that the occurrence of severe COVID. And so that would be, for example, 150 compared to 75 cases in the two arms. So that's kind of where that comes from. And I bring that up because you hear people talking about, what about these early looks? You know, the president and some others are saying, well, we're going to take a quick look, and maybe the first look at the vaccine might give us some clues. And indeed, in studies, both because of efficacy and because of safety, there are interim looks planned. And for example, they might, often with these COVID vaccines, they're looking after a third of the cases, roughly a third of the cases might occur. And so if the placebo arm, for example, has had a third, let's say that would be 53 cases, then the other arm with the vaccine would have to have very few. And so let's say that happens and it's successful, but it's successful with the first vaccine. What would you do if all of what we know about the COVID vaccine in the world is based on the difference between 53 people getting it versus 25, for example? And that's kind of, and then we don't know whether it's the older elderly, or we don't know whether it's women versus men. We don't know whether, so there'd be a lot of questions left if we have very sparse data like that. And yet that actually is possible given the designs and given the planned interim evaluations, that we might get that look. And yet there's the ethical question of, well, if we know it's working already because it prevented infection in the first 53 instances, then shouldn't we use that information? Shouldn't we start to approve the vaccine? Shouldn't we stop all the other studies and put people on the one that's working? Those are the kinds of things, just to give you a sense of where we're headed as a country. Those are going to be the things you're going to see about in the papers. And then, of course, there's this issue of like, can you just bypass the whole regulatory system? You know that in China and Russia, they're giving vaccines to at least certain populations without full phase 3 approval showing efficacy. And then there's the EUA, the Expanded Access Program, that has been used in the US in the times of the pandemic, that can be used to bypass, and is fortunately for this vaccine not being deployed. Okay, so all of those sort of preliminary comments then kind of converge on these. By the way, this is a really great site. If you just want to have one place to go look to keep up on the COVID vaccine, this one in the New York Times I like and use myself. And so this is kind of where we stand right now with a number of vaccines and in different stages of development. Okay, so even 11 of them already into this late phase 3 kind of work. So a lot of progress. And when people say things like shots on goal, that's what they're talking about, like 11 shots on goal already. So I'm not going to talk about them all, but I am going to illustrate using some of the ones that were involved in the Operation Warp Speed, and that you might be reading about and may even be getting, some of you. And they're kind of in 3 groups mostly. And one group is these ones called the RNA vaccines, okay? And what the reason that these were the first ones that were made is that these things, all you need to know is the sequence of the microbe, or the part of the microbe that you're trying to protect against. Once you know the sequence of it, you can make the RNA, which is sort of like the analog of the DNA. The way that the DNA gets certain proteins made is through RNA. So they're bypassing putting the germ in the body, and they're just making the RNA, and then the RNA would go into cells, and then the cells would make the right proteins, and then the body would respond with immune responses to that. And so they picked these spike proteins, and they made the sequences really just within weeks of the Chinese releasing the viral sequence. And this company, Moderna, has been doing this for years for different conditions, unsuccessfully. But then as soon as they saw this sequence, they made the RNA, and they were the first to put their vaccine into humans way back in March, and demonstrated that it works in animals. Now, these RNA vaccines have this attribute of, first of all, they hurt. They do hurt when you put them in. You have to give them twice, and the second shot especially hurts. And they have to be frozen. They have to be frozen at -80. So not even the kind of freezer that you have at home, but a really deep freeze. And so that presents sort of a complicated situation for their long-term distribution. And when you think about sub-Saharan Africa and parts of the world where that could be challenging. But nonetheless, these are among the first. These are the forerunners. The Pfizer partnered with this German company. And so these guys are way out in front. Well, they're a month in front, and will be some of the first vaccines that you will hear results of in the press. Okay, 2 shots separated by about a month. I think it's 21 days. Yeah, 21 days for the Pfizer, 28 days for the Moderna, and late phase 3 already. Lots of progress. Another way to create immunity is to take another virus, in this case, something called an adenovirus, or a serious cold virus, and to stick the virus, the part of the SARS that you want, into the cold virus, and then put that into people. And then that virus does all the work that normally occurs with a virus, and tricks the human cells into making immunity against SARS. And so we've used these kind of vaccines for years for different kind of conditions, including very serious conditions like Ebola. And so one of the common ones are these adeno. You hear people say chimp ad, or ad5, or ad26. They're referring to an adenovirus, and the number of the virus refers to just different strains of it that have been kind of weakened in the laboratory so they don't harm anyone. And then inserted into it is the important part of the SARS virus. So these studies are also part of the Warp Speed, and they're 30,000 person kind of viral vector. You've heard that the Janssen is notable in that that this one is a 1-dose vaccine with a human adenovirus. The AstraZeneca or Oxford one that you've heard about is a 2-doser, and it uses a chimp adenovirus to get around worrying about maybe there's already immunity to the human adenoviruses. So this adenovirus comes from a chimp. No humans have seen it, and so it's good for vaccines. These are up and running all around the world, and will be close behind. And then a third strategy is the idea of just taking apart, making the protein. So instead of giving the RNA to get the cell to make the protein, or getting another virus to stick it in, here you're just getting, you're making the protein in the laboratory. You're formulating it specially to make an immune response against it using things called adjuvants and lipid nanoformulations and that sort of thing. And so multiple companies are trying this. This is, for example, what's used for the hepatitis B vaccine, the first recombinant protein-based vaccine. So these are ongoing, and here's some examples. A Maryland-based company, Novavax, and then Sanofi GSK, and there are many others with these. So those are examples. There's also something called formalin inactivation, various inactivation techniques where they actually take the whole virus and then put that in. There's some others where they're taking not just that outer protein, that spike, but they're also betting on, well, maybe if that's wrong, we'll put another one in too, and putting a couple proteins in to hedge against what might be successful. And all of these are trying to create the good kind of immunity, not the bad kind that puts people in the intensive care. But we don't understand that immunity so well right now, and that's one of the reasons for pause. So this is a slide that I stole from Asif Slawi, who's running the program, the Warp Speed program, and just to give a sense of the kinetics of this and where the various projects are and when they're expected to finish phase 3. And then you see that 50, 100, 150 with the Moderna. That comes back to this point that I was making where there will be this first sneak peek of 50, okay? And that's what you're gonna probably read about pretty soon. And that's why I kind of spent a lot of time setting that up so you hopefully understand that. But it will be on a very few number of people, and the study was initially designed for 150 outcomes in the placebo arm. So we'll see. And what if COVID like sort of goes away in the towns where you're doing the vaccine? Well, then it might take longer. It might take more than 15,000 people in each arm. And so the J&J approach is to recruit more. And so there's that kind of thing. Those are some of the dynamics that we're dealing with. And so, yeah, I finished with this slide, which is there will be issues. For sure, there will be issues. I've tried to underscore some of them as I've gone along, like this issue of first and success. So if with just knocking 50 from one arm down to 25 in the other arm, are we going to stop this whole thing? And at that point, so that's a big question. Or are we going to keep stating it differently with a different tone that may make you lean in another direction? Would we keep doing things that are experimental when we know one thing works? That's another way of looking at it. So that creates a lot of interesting discussions that you'll be reading about and maybe participating in. There's the issues of access, of course, trying to make sure that people all around the world, nothing like an infectious disease, as Leonard pointed out, underscores the importance of how we're united in the world. One person's health is related to another's. And Bill Bishai is an expert on tuberculosis and has been preaching this for years. If one person has TB, it makes everyone at risk of TB. So you can even be selfish and care about global health when it comes to these kind of understanding from these pandemics. And so there's other issues with how will we make enough of them, and then these questions that I've already gotten into. But I'd rather wait and take my questions at the very end. I will. This is a couple weeks ago. There was that weekend when there were like all these storms converging, and no one wanted to forecast which one was going to hit the United States. And so I took this picture because I don't want to forecast exactly what's going to happen. I would be wrong. I predict that, but I want to let Bill have some time to talk about his exciting scientific developments with his work. So Bill, I'm going to hand off to you
now. Thank you, Dave, and good morning, everybody. So one of the vaccines that was on Dave's list of 11 in phase 3 turns out to be a vaccine that is 99 years old, the vaccine called BCG. And so what I want to talk about in just a brief 5 to 7 minutes is the concept of repurposing an existing vaccine as a way to contribute to immunity against COVID-19. Essentially ask the question, have we actually had a diamond in our back pocket without fully realizing it? I have one disclosure, that is some work that I'm doing with a novel recombinant BCG strain, but I'm not going to discuss any of that technology this morning. So what we learned in grade school or from our pediatrician was, I'm going to give you this tetanus shot to prevent tetanus. You're going to get your flu vaccine to prevent influenza. This diphtheria shot will keep you from getting diphtheria. One vaccine for one disease. But is this really true? And I want to give you 3 anecdotes before moving on to COVID about the concept of non-specific immunity. That is, do certain exposures or even vaccines that we receive give us broad-based immunity against things that they weren't originally intended to protect us against? So my first example goes back to 1893, where bacteria were used to prevent or treat cancer. A fellow named William Coley at the Hospital for Special Surgery in New York developed these mixtures of bacteria, including the strep throat bacteria and a serious gram-negative bacteria that he would mix together and inject into tumors in patients that he treated by the hundreds. And he achieved quite a bit of acclaim in the day. There, as you can see on the right-hand side of the slide, this gentleman had a very serious facial tumor, and after several injections of the Coley mix or Coley's toxins, his cancer seemed to resolve. It was never put forward in controlled clinical trials of the type that Dave was telling us about, but nevertheless, the product sold for many years and was on the market until as recently as 1963. But the take-home message here was that bacteria that have no relationship to cancer gave us an immunologic response against totally unrelated tumor cells. My second example also goes back to the 1880s, where a fellow named Julius Wagner Haureg from Austria came up with the idea of giving patients who had serious forms of syphilis, neurosyphilis, which made people crazy and had a very short survival time. It was known at the time that high fevers prevented malaria in animal models. And so he hit on the idea of exposing these patients to true malaria. He would take blood from a malaria patient and inject it into someone with a serious form of syphilis. And it was called malariotherapy. And it did, in fact, help some patients. Others were hurt. And in fact, there were some deaths due to the malaria. Nevertheless, it was considered state-of-the-art. And Professor Wagner Haureg won the Nobel Prize in 1927. Take-home message here was that one microbe, in this case malaria, elicited an immunologic response against a totally unrelated microbe, namely syphilis. Now to the organism that may actually be of value against COVID-19, Bacille calmette-guerin, which is abbreviated BCG. It's about to celebrate its 100th birthday next year. These two French microbiologists, Albert Calmette and Camille Guerin, beginning in 1908, started taking virulent tuberculosis bacteria and just serially plating them on a new plate once a month. And they did that about 900 times over 13 years. And the bugs started to look different on the plates about halfway through. And in 1921, without any real clinical trial, they decided that it might work. So they began vaccinating children at high risk in Paris. And in fact, they did prevent tuberculosis. So this was a live attenuated bacterium closely related to the TB microbe itself. And since 1921, BCG has gone global. It's believed to work in 60 to 80% effectiveness rates, particularly for children. And now, according to WHO guidelines, it's given to all babies at birth, except in the United States, Canada, and Western Europe. And here you see on the right-hand side, a newborn infant getting BCG vaccination. But a very interesting observation came along about 50 years later, and that was in patients suffering from bladder cancer, if they happen to be exposed to BCG in the bladder, their bladder cancer resolved. And this has revolutionized non-muscle invasive bladder cancer. And in fact, for patients unfortunate enough to have that form of the disease, BCG is curative about 70% of the time, given into the bladder repeatedly. So the third example is this is a bacterium that elicits an immunologic response against totally unrelated tumor cells. Again, raising the question, can exposure to an immunologic challenge give broad-based immunity rather than specific immunity? So it's a very hot topic in my field, the so-called idea of non-specific immunity. Dave talked about training the immune system, and this phenomenon that I'm talking about is also a form of training, but it's a non-specific training. After a first challenge, the immune system responds, and it's trained during this interval of time, and at the second challenge with a totally unrelated organism, the trained response is even greater than the first response. And Professor Natia over in Nijmegen, Holland, has really written the book on this. He has shown through studies of his own as well as reviews of the literature that children who have been vaccinated with BCG at birth show lower rates of human papillomavirus causing warts, respiratory syncytial virus, influenza A, and herpes virus. So very curiously, by looking at what has happened in cohorts of patients treated for tuberculosis, we've observed lower rates of these viral infections. And in fact, Natia himself did a study prospectively showing that prior exposure to BCG helped lower the ability of yellow fever virus, a live vaccine, to proliferate in humans. So he called this phenomenon trained immunity. And some interesting advances have been made scientifically. It's thought to be epigenetic reprogramming, where the genes are not changed, but the exposure of the genes for use in the body are opened up, and that's shown over here on the right. Closed chromatin is tightly wound, and an epigenetic change can be elicited by BCG opening it up, allowing our cells to make more of the particular genetic products from those open areas, which tend, in this case, to be genes required for immunity. And so with the knowledge that BCG worked for RSV, influenza, and herpes simplex, the question was raised, would it work for SARS-CoV-2? And that question began to be answered very early in the pandemic. Beginning in early April, studies launched giving BCG to healthcare workers and other patients in high-risk groups in numerous countries, mostly in Europe. So we observed the emergence of the pathogen, at least in our country, in March of 2020. The clinical trials for trained immunity inducers have started. We're now out about 8 months, and we expect to begin to get results depending on the density of COVID-19 in the areas where BCG is being studied. With any luck, the resurgence, as frightening as it is, could accelerate the endpoints of these studies, just as Dave mentioned. And maybe we will know if BCG works for COVID-19 as early as January 2021. Another very interesting approach has been, well, maybe if one of the specific vaccines that Dave told us about is, say, 50% effective, and we know we need to get to 75 to have herd immunity, maybe BCG would be something we would want to combine with one of the specific vaccines as a booster. So to summarize, the one disease, one vaccine may not be strictly true. The TB vaccine, BCG, gives elevated non-specific immunity that's been known for a long time. And so this is an example of repurposing a vaccine that we've had on hand. And as Warp Speed develops specific vaccines, BCG may be something to keep in mind as a booster for those specific vaccines to achieve herd immunity. So I'll stop there.
Okay, great. Thank you so much for the presentation. We already do have a few questions coming in, so I'm going to read them out, and I will see if Dr. Thomas can join us on video again. And so I'll go ahead and ask that first question. And actually, we have a first starter off question. Leonard has a question to get us started, so he's going to join us, and then we'll get started with the other questions.
Bill, there's a question about why we don't use BCG in the United States.
Yeah, the United States has really done amazingly well in keeping tuberculosis rates extremely low. And so BCG, when it's given, takes away one of the diagnostic tests, the tuberculin skin test that many of you may have had. And so it was thought back in the 70s and 80s best to take advantage of that important diagnostic, since we don't have very much TB anyway. There's not that much to prevent.
Gentlemen, thank you very much. I would like to ask just a general question. What about this virus, this new virus, has surprised you the most, and what has not surprised you the most? And whether about the virus or our reaction to it, I'm curious as to how each of you see that question.
Go ahead, Bill.
Well, I was surprised during my time taking care of COVID patients here at Johns Hopkins by the enormous variability in the disease. We had a lot of patients that I cared for who happened to have COVID, but were in the hospital for a skin infection or appendicitis. And then we had the ones that were desperately ill with the severe lung infection. And of course, we know that there are plenty of people out there with totally asymptomatic COVID infection. So the variability in the human response has surprised me.
Yeah, no, it has been shocking, the differences among people. That's not that one person can not even know they had it, and another person, even a young person, it can be fatal. So, and the range in between, and that we don't really understand why. I think that surprises me. I think we all expected this to occur, but we thought it was going to be an influenza virus. Meaning like a lot of people, a lot of infectious disease people were saying there's going to be a pandemic, and people have been talking about this for decades, and there's been lots of meetings about it and roadmaps and ways to plan, and everyone thought it was going to be an influenza virus. So I think that when this was originally described as a corona, we didn't have the vigor. It was sort of a little bit of a yawn that didn't last long, but as a global infectious disease society, I think we were all waiting for flu to do this, and then a coronavirus did, and that surprised me.
And just a quick follow-up, what does this variability in human reaction to the virus maybe suggest to the two of you about the nature of the virus itself?
Well, I'll just
sorry, go ahead, Bill.
The reason I went into infectious diseases is because the bugs know more about human biology than the doctors and scientists do. And there's something that corona knows about us that we need to discover, and that's going to lead to a biomarker. But at this point in time, Lenny, I couldn't tell you what that marker is. Dave?
Well, it's interesting. Infectious diseases, more than any other medical condition, differ person to person based on their host genetics. Okay, so even more, you know that some cancers run in families, and you know that heart disease runs in the family, but in a much greater sense, the outcome of an infectious disease is determined by our DNA. And so if you take the DNA of people who have severe COVID and those that don't, there are some differences that have already been recognized. And those may in time give us some clues as to why some people do much worse than others. But right now, even that kind of research is just very preliminary.
Okay, and there's some other questions that have come through. I can read them out to you all, and whoever wants to pick it up can run with it. The first question is, why is BCG not given to infants in the US, Canada, and the EU, and do you think it should?
So I touched on that at the beginning. When that decision was made, we relied on TB skin test to diagnose TB, but if you give someone a BCG vaccine, the skin test will turn positive. And so it takes away our ability to diagnose latent TB. Now we have better diagnostics, and it's possible that that decision will be reconsidered. But I will say that United States is controlling tuberculosis extremely well. We have about 20 cases a year here in Baltimore, and overall it's got a very low incidence in the United States. So there doesn't seem to be a strong need to give BCG, even though we don't have that diagnostic requirement as we did in the 70s.
Okay, great, thank you. Another question has come in. Which of the vaccines are not live?
So the ones that I mentioned were like a protein, or the ones where they're inactivated, and even the one that the very first group I showed that were based on RNA, or in some cases DNA, those aren't live. Okay, so they don't have any. The ones that are live, it's easier to talk about that, are the ones that use these laboratory weakened viruses to bring the SARS-CoV into the human cells. So those are the adenovirus ones, and there's a VSV one, and actually there's one now with rabies. But I think the marketing department of that company is starting to quit. So I don't think that one's going to go too far.
Okay, great. Some more questions have come in. It says, I have read news reports that the coronavirus 19 virus is mutating in response to mask wearing and social distancing tactics. How worried about this are you?
Bill, we're looking at each other to see who's going to field it. I'll just say, so far there are some differences in some of the strains of COVID that have been found around the world. But so far, we don't have any reason to think that those have been produced by mask wearing. And so far, we don't even think that they've occurred in a way that is going to make them more harmful, or possibly cause a reinfection of somebody that already formed an immune response, let's say, to an earlier strain. But those are important questions, and people will have to keep an eye on that, because of course with influenza, that's the whole thing, right? That's why we're having to redo it all the time and get a flu shot every year. Is not just because the immunity that's produced is not very strong and long lasting, but it's also, and even more the case, that the virus shifts on us. So we're keeping a close eye on that one. But so far, in the really important parts of the virus, there's not much evidence that it's shifted.
Yes.
Okay, great. Next question. The Germans are starting a vaccine use before the end of the year, claims news reports this week. Do you know their approach?
I mean, the Germans are involved in multiple studies that are going on throughout Europe, but I don't know which one they're referring to that started a vaccine use before the end of the year. So that may be really pretty much in the same kind of time frame that much of Western Europe and the United States is expecting to have an approved vaccine. Now in China and Russia, there's unapproved vaccines that are being used that haven't finished phase 3 studies.
Okay, another question. So we're going to keep going with the questions. We've got a few more. It says, we were lucky that COVID-19 was part of a well-studied set of viruses. How worried are you about a really new, previously unstudied virus, like from the melting Arctic?
Bill, do you want to do that one? You do the Arctic virus.
You know, that one of the things that we learned from this epidemic and then the one in 2003 called SARS-CoV-1 was that there was a jump from animals to humans. And there has been a strong effort in the United States funded by the National Institutes of Health to survey viruses of animals for potential jumps. We have not really delved down into places on Earth where not many humans go, and there is a remote possibility that pathogens could exist in the permafrost, for example. But our focus in the United States has been on viruses that are out there already and animal hosts that could jump over to humans.
Okay, next question. And I'm going to combine these two questions. They're very similar. How do you socialize? Would you host friends in your home without a mask? And the other one was, just out of curiosity, do the two of you go out to restaurants at this point?
Well, I'll start. I don't go to restaurants. We have a quarantine group that includes my immediate family, and when we want to interact with people outside that group, we have taken advantage of the warm weather and done it outside, and done it with distancing and mask wearing. So that's an honest answer. What will happen at Thanksgiving? I haven't thought through that yet.
I have a 4-year-old. We have a bubble, my wife and I and my 4-year-old. Having a 4-year-old means he has friends, and so we do things outdoors with his friends that he attends school with, and we have dined outdoors. And again, like Dave, we're not sure what to do about Thanksgiving.
Great, thank you. 2 more questions so far, and then, okay, so we'll see. Do you believe that the huge number of American deaths indicates an unusually low health condition for hundreds of thousands of us? Should Americans generally have reflected more robust health and immune strength?
Yeah, I mean, it's very disappointing, right? To be on the highest end of mortality with this. We have the most expensive healthcare in the world, and it's disappointing when it's not making us healthier. And so that's a big problem. And to be sure, our population, the weight, the obesity issues, and some of the other chronic medical conditions that are prevalent in the United States contribute. But yeah, I mean, I think that's that I get that question. I think it is one that makes a lot of people concerned. That the sad thing is that I think Bill and I both would have bet that by now we would have been more confident and more in that we had more therapeutic tools. And it's kind of been a little bit disappointing that something like steroids is kind of the big hit this far into it. Something that's been around forever and is not that sexy of a treatment. But that's the one that improves mortality at this point. So, and that also may sort of explain why in other parts of the world they're doing a lot of what they can do. Same things as what we're doing here.
Okay, great. And then one final question, and I think Leonard wants to jump back in after this. The final question is there any data on the results of the BCG vaccine administration to healthcare workers in Europe?
So unfortunately, the studies are all listed as accruing, but none have reported data as of yet. There was one study that started a year and a half ago before COVID, looking at the efficacy of BCG in elderly individuals. And it was opened and published early because of COVID over questions of whether BCG is safe. And so even though it hadn't gotten to the final point, the 150 that as Dave was talking about, they opened it up. And interestingly, they found that individuals over the age of 65 who had been given BCG had fewer infections statistically significantly than those who received placebo. So it was and there was no issue with safety. But that doesn't tell us if BCG will work for COVID. It suggests BCG prevents a whole bunch of things, and hopefully it will also prevent COVID.
Gentlemen, thank you very much for your time today. If there are any other questions, get them to us and we will get them to our panelists. I would like to ask you to let us know, we are a financial advisory firm, and so we, by definition, have an audience today of people who have money, many of whom are charitable. Is there some vehicle at Johns Hopkins if people are so moved to contribute to the research that is going on there? Could you let us know about that right now? What are some of the more charitable giving arms at Johns Hopkins that our audience should know about?
I was prepared for everything but that. I guess that's why we went into infectious disease. We're destined to not. Yeah, well, there are a number of issues related to COVID and not. And Bill has some innovations that he's been developing that in his own lab. So I think, Leonard, probably the easiest thing is to talk with someone about that if they want to, and to understand the kind of things that they're interested in, and then to come up with a proposal that matches the kinds of interests that someone has. Our websites are full of ideas and different programs that are targeted from COVID all the way to TB and other cancer and other conditions. So I'd be happy to talk with someone about
Great.
that, and I'm
so
sure would too.
Any of our clients today or guests who are on the call, if you are interested additionally in Johns Hopkins and their research and how you can be helpful to them, let us know, and then we will get something to David and Bill. Thank you both so much. We are super, super grateful. And I learned so much listening to you today. And we're so thankful for your expertise and your knowledge in helping us and our clients really get stabilized in foundational information that is so helpful to us. So thanks to both of you. Hope you have a great weekend. Everybody, we're going to take about a 5-minute break. No, a 5-minute break. Definitely a 5-minute break. And we will be back to start up around 11:35 Central Time, maybe 11:36, and I will be doing my presentation then. Back in just a few minutes. Thank you.